My name is Enrique Antonio Dominguez Menses. I am life sciences, and this is my BlueSky proposal. So one of the big challenges we face today in medicine is dealing with cancer, and that's very difficult because there are many different types of cancers, and there's various symptoms, and there isn't one cure that could, be potentially used to help all the patients dealing with all the different types of cancers. So my idea or my conception of the ideal drug to deal with cancer would be one that's A, affordable, and B, that's not tailored to individuals. Today we see unfortunately, because of the advent of epigenetics and specific genes for people, we see that drugs are becoming more and more tailored for the individual, and this results in higher costs. So although we are getting closer to solving the problem for individuals, it is unlikely that this will be affordable, for the mass, for the people, for the common citizen. So what my idea was to have an approach that you could mass produce and you could use to cure different types of cancers. So here is the cell, and here are the receptors that we learned exist in LS1a. Cells have different receptors, and in cancerous cells, what happens is the receptors are constitutively active. They're in the constitutively active form, causing the cell to continually proliferate. Now, this is not for all types of cancers, but most types of cancers have constitutively active forms of receptors. Naturally, though, or regularly, you would have a ligand bound to a receptor, which causes the two receptors to dimerize and tells the cell to proliferate. However, in a cancerous cell, the ligand is not there, and yet the cell has the constitutively active form of the receptors and is continually proliferating. So this was a window or an opportunity to target these receptors. Now, a problem with cancer that I mentioned is that there are many different types of receptors because there are different types of cancerous cells. So instead of targeting different types of receptors, you could target the idea of constitutively active forms of receptors. And what makes that promising is that there is no ligand bound to constitutively active form of the receptors in cancerous cells. So this is what I thought was a window, to target in order to treat cancer. Now, to treat cancer, you in my proposal, you would use a virus, a lentivirus. This lentivirus would have an expression vector with DTA, the diphtheria toxin A. Now, the end goal of this proposal is to have a lentivirus identify cells with a constitutively active form of the receptor and transfect those cells with DTA. If the cells express this DTA, they are ablated. It's a toxin, so the cells die. So this would effectively terminate any cancer cells in the body. Now, the problem is targeting these cells. What we learned in LS1a is that cells have receptors. Now, a way that the body deals with with targeting cells or labeling cells is using antibodies. Now, I will label this antibody number 1 and this antibody number 2. So to deal with the fact that there are different types of receptors, you would have to use a system in which the lentivirus can identify different types of receptors. Now, a possible approach to do this is to use a system in which you have a primary antibody and a secondary antibody. The receptors, the constitutively active form of the receptors, act as a primary antibody. Now, you could engineer the secondary antibody to label the receptors. So in this diagram right here, the second antibody would attach to the constitutively active form of the receptors and be ready to be recognized by the lentivirus. What this allows for then is for a virus to have multiple secondary antibodies that can identify that one second antibody that's attached to the receptor of the cell. So in this approach, what this allows for is a mass production of one virus to treat different types of cancers. And it's all based not on a specific receptor, but the idea that there is no ligand bound to constitutively active receptors. What happens if you express DTA in non-cancerous cells? In other words, there's a risk that you might express DTA in perfectly healthy cells and be killing the patient. Now, you could argue that it's, chemotherapy does that as well, but what I try to do, in my BlueSky proposal was, one, an approach that was affordable and non-tailored to specific individuals, and two, an approach that's non-deleterious, to the overall health of the individual. So to overcome this, you could modify the expression vector of the lentivirus to be driven by specific promoters. For example, you could use the TEP promoter. And then what this does is it allows you to control the expression of DTA based on a compound such as doxycycline. So in a theoretical approach, what you would do is you would feed the patient doxycycline and observe whether or not cancerous cells are being ablated and whether or not healthy cells are being ablated. And depending on the results, you might stop the treatment and prevent the ablation of healthy cells. In the end, I think it's a feasible approach. I read a research article in which they caused the ablation of specific cells in mice using a similar method, however, driven by the Cree system expressing DTA, and they were able to ablate specific cells in mice. So this makes me believe that this is possible for humans, and it could be a viable approach to cure different types of cancers. And that's my BlueSky project.